Dear students, welcome to our lecture on bioinformatics. So in this part of our course, Applied Environmental Microbiology, we will see how we can use tools from computer science and mathematics to answer and make sense of the data that we generate using biotechnological and microbiological assays. In one of the previous lectures, I have talked about sequencing techniques, all the first generation, the second generation, the third generation and the fourth generation. From the third generation and fourth generation sequencing techniques, we generate tremendous amount of data, and that tremendous amount of data is not something we can analyze using our Excel sheet. And while we are at it, I must also mention, even from second generation techniques, we generate enough data that we require sophisticated tools that can help us make sense out of the data. So it is not just the sheer amount of data that we generate. For example, if you do metagenomics, we might generate anywhere from 60 gigabits of data to 120, 200 gigabits, which implies millions of base pairs per sample information would be provided to you. So that is a lot of data. So it's not, and in addition to it being big data, we have other hurdles too. For example, in our sequencing techniques, regardless of which technique we use, there will be sequencing errors that will come into picture. There will be other chimeras that would be formed and sequenced falsely, leading to false positives and confusion. Also, with the data set, you need to align it to the existing databases, and all this cannot be done manually. We need algorithms and programs to do that. For that reason, biology has collaborated with computer science and mathematics to come up with bioinformatics, and that is what we will talk about briefly today. So bioinformatics, as the word suggests, is bioinformatics. So we are trying to get information from the data collected from biological studies. Now, as environmental engineers, what would our environmental scientists, environmental students, or students of this course, Applied Environmental Microbiology, for us typically the most important big data generating technique that we use is sequencing. So we can refer to it as metagenomics. If you remember about metagenomics, one of the key features of metagenomics is the ability to sequence genes, to sequence genome and extra chromosomal material too, genetic material too, from microbes that cannot be cultured. So without culturing them, so we skip the culturing method, culturing step, and we directly sequence or sequence them after amplifying the genetic material. Now in metagenomics, what will happen is we will get a good snapshot of who is present in our sample and perhaps even what they are doing. So we can do functional metagenomics that will give us more clear information on the functionality of the microbial community. Either case, metagenomics is very useful for environmental engineers, environmental scientists and researchers, and metagenomics, as name suggests, generates tremendous amount of data which we cannot make sense of without bioinformatics. Alrighty, so now look at metagenomics here, and briefly I have talked about this earlier in one of the lectures of this course, that when sequencing techniques were developed, we finally managed to sequence the entire human genome, which was a big achievement. It was not done single-handedly by one research lab, but was a collaboration between many PIs and many researchers. Now as such, you should get an idea about how difficult it is for us to sequence and make sense out of environmental samples using this code by Craig Venter. The soil work is like trying to assemble 40,000 human genomes simultaneously. So and if you have not already done so, go ahead and do it. Look up human genome sequencing online, find out about the scientists who were involved in it and what kind of supercomputing tools were required, how much time did it take, and what you will find out that it required a lot of computational facility, because human genome is a eukaryotic genome. It has a lot of introns, exons and other genetic materials that are not present in prokaryotes like bacteria, which usually tend to interest us more from environmental perspective. So when we are sequencing human genome, not only we need to have the sequencing part done, but then we need to get rid of the errors or suspected errors. We also need to have an idea of the quality of the sequences we are generating. We need to get rid of chimeras, and after we have done all the QA/QC work, next step is to assemble them. So let's look at briefly about what assembly is. So when we sequence, and this is shotgun sequencing, you have a, let's say, a bacterial genome, so it is circular, and because we are doing shotgun sequencing, what we do is we split it into small pieces and we get small sequences fragment. Now one singular genome can be split into, let's say, 5,000 pieces, 5,000 fragments. Now each of the fragments will be sequenced by your sequencer, and once you have the information, the small sequence, the small length, that the small fragment of your genetic material may not be as informative as the entire genetic material, because see, many things are happening in our genetic material, whether it is plasmid or chromosomal genetic material. The sequence of genes is important, the location of genes is important. Also, if sometimes when we do shotgun sequencing, not sometimes, many a times when we do shotgun sequencing, we don't get the entire gene in one sequence, they get split up. So we need to assemble so that we remove all confusion, we gain clarity, and we know the sequence of genes in the genetic material. So for that, it is very important to stitch back these fragments. So now you have generated this data, and it is important to stitch them back. So stitching them back would look like this. Let us say I have a sequence here, and here it has a particular sequence that I find pretty unique, and then I find another sequence that has similar or exactly same sequence on and in the right orientation. So what I can do is I can assume that because of the similarity, there is a good probability that they are actually in continuation with each other, but there was a break here for one bacteria or one genome, and for another there was a break here during shotgun process, and thus we have this split. So what we can do is we can stitch them together, and now we have this, a longer fragment. So what we can do is we can take our sequences and assemble them, and assemble these longer fragments further. For example, they have unique signature here, there might be another unique signature here, and if we find another sequence or another contig that has same sequence here, so we can stitch them together, and this way we keep growing longer and longer sequences until we reach scaffold level. So we have scaffolds really long, up to 1000 base pair and more. So these are more meaningful because they typically would have gene sequences. You will be able to know, okay, gene 1 appears before gene 2, appears before gene 3, and it is in the right according to the orientation, or in another sequence. So and then the scaffolds can be further assembled together to recreate the entire genome. So this is what we call about genome as sequencing the genome. The bare sequencing is the first step of recreating the entire genome, getting the information from the entire genome. So alright, I have a 4th generation, 3rd generation sequencing machine, and it gives me this data, but I still need to go through all this step, and these steps are called assembly. This is assembly process, basically stitching the smaller sequences together, so we have a bigger picture, broader picture, and ideally we want to go for whole genome sequence. So we get the sequence for whole genome, we can assemble it, and we can stitch them together, and now we can say, let us say this is bacteria 1, bacteria 1 has been sequenced. Or if this was a human cell, so we can say the human genome has been sequenced, right. So this is computationally very challenging. So we have algorithms which decide similarity, which stitch together, which check the error rate, because every time you are stitching together sequences, we are losing our certainty about the sequences. So we are adding to error. So for that we have algorithms which decide whether it is worth stitching 2 sequences together or not. Another thing I want to say, that here in this example that I gave you, notice that I have only stitched one sequence with the other sequence. Typically in metagenomics, what you will have is you will create multiple copies of same fragment. So let us say this is fragment 1 from one bacteria, this is fragment 2 from second bacteria. They have some similarity, but there will not be just one sequence or one fragment that would be like this, because sequencing techniques often involve amplification step. We will have multiple copies of first bacteria's fragment 1, so multiple copies, and the more copies we have that agree with each other, that yes, all these copies are very similar, so we can say that they are copies of the same fragment, they are same size, they have similar sequences, and And if all of them, if two of them have same match, so fragment 1 and fragment 2 both have same match, no matter how many sequences we have that we can label as fragment 1 and fragment 2, then we can with confidence stitch them together. So this is the importance of the word depth. So so when you are and that brings to me to another point, that when we are doing metagenomics or sequencing metagenomics, basically you need to know number of base pairs that you will generate. This will give you an idea of the width, how much data are you going to create. Obviously the more is better. Next you want to know about the depth, which is basically one single fragment. Let's say in my sample I have one bacteria that is unique and it's only one of its kind, and I split it into fragments in the shotgun process. So this one single fragment, how many times will it be sequenced, or how many copies of this sequence will I get? This is very important, the depth. And depth is very important because when we are assembling, the more the depth we are, the more sure we can be about our assembly process. Note, the depth will depend on many things. Depends on your platform that you are using. You know, some platforms give you good depth, some don't. It depends on the diversity. If you have a lot of microbes, then the depth would not be as much. Or if you have pure culture, then definitely you have very good depth. The other thing it depends upon is GC content, because as GC content changes, the annealing, this annealing step in the sequencing process gets affected. So there are multiple factors that will determine depth, but depth is very important. Alrighty, now let's come back to the quote by Craig Venter. Here he is mentioning the soil work is like trying to assemble 40,000 human genomes simultaneously. So he is trying to explain to you, this was very difficult, getting a human genome assembled together, stitched together. We had to use high performance computing in USA, perhaps the best in the country there, and one of the best in the world definitely. Now imagine 40,000 times over. That is the challenge for soil microbiome. And I hope this also gives you an appreciation of the kind of complex microbial communities we are dealing with here. So one human cell, nowhere near as complex, nowhere near as diverse as soil microbial community. And let's continue listening to Venter. He says, I think this is an area, and he's talking about metagenomics and sequencing and including bioinformatics, that will have far greater impact on science in sequencing the human genome. Which area? Sequencing the soil microbiome. Says Venter, not everyone might want to go quite that far, but Sherper is happy to insist. This is really a new era in microbial ecology. Alrighty, now let's look at the hurdles for ecological studies and how metagenomics is a promising feature and why you would perhaps want to use it and why that will push you towards bioinformatics. So, in ecological studies, and I hope you have learned this, that microbial communities are not static. For example, if I have a wastewater treatment plant, I do its characterization and I tell you, well, in my wastewater treatment plant I have these protozoa, I have these bacteria, I have these fungi, I have these bacteriophages. This is how the microbial community looks like, and yeah, I know how it will behave because I know the community structure, I know the functionality. But hopefully by this time in this course, you know that microbial communities are not static, they are constantly evolving. So they are constantly undergoing succession for many reasons. One is the random error that crops in, and then the random deletion of a population or addition of a population. Next is that influx and efflux of different kinds of samples and inocula that change the microbial community. And yet the most important is that the environment of the sample changes over time. For example, we have talked about lakes, if you remember. So at the bottom of the lake, no phototrophy, definitely, because a lake does not, in that level, lake does not receive sunlight, and most likely if it is a deep lake, it is anoxic or anaerobic, and in this case aerobic microbes cannot thrive. So where would we expect aerobic heterotrophs, where would we expect autophototrophs? At the top of the lake. Now as oxygen gets depleted from the bottom, because there are oxygen consuming reactions happening, and which is basically oxidation of food, oxidation of electron donors, and when that happens, oxygen gets depleted and we start getting a redox gradient in the lake. Depending on the stratification of the lake, we will get a redox gradient. Now that redox gradient also does not remain static, it too changes. It too changes with time as oxygen keeps on getting depleted, or if reaeration is well enough and it starts entering into the lake, and then there are seasonal patterns. So the take home message here is that ecology is not static, biology is not static, definitely environmental microbiology is not static. And a very clear cut example is that for 15 years your wastewater treatment plant worked perfectly, no problems, microbes are happy, you are happy with the microbes and the work that they are doing, and then one day sludge refuses to float. Why? Because microbial community has undergone a succession. So this is a major this is a great point for appreciating environmental studies, and yet it is a major hurdle. Why? Because let us say I took a sample from Delhi landfill on September 24th, alrighty. Let us say I took this sample from Delhi landfill, and then I characterized the microbial community. I said, okay, species 1 these present, species 2 these present, 3 these present, 4 these present, so on and so forth. And I did all kind of analyses, alpha diversity, I compare, and then this was on September 24th, 2017. And then I went to, let's go to Mumbai. I went to Mumbai and I collected sample from Mumbai landfill on October the 1st. And then obviously I got information, species 1 so much, species 2 so much, species n so much, right. Now the question is, can you compare Delhi landfill with Mumbai landfill? For example, let us say I want to publish a paper comparing microbial communities from different landfill, different demography, different region, different geography, different climate, and then I do this. I have sequences for microbial community in Delhi landfill, I have sequences for microbial community in Mumbai landfill, and when I compare, the question is, how much similarity are you expecting? How much differences are you expecting? Are the differences only because this is Delhi and that is Mumbai, or are the differences according to the age, according to the time? This was September 24, that was October 1, right? So maybe there was a rainfall event, or maybe geography, climate. So there are many variables that are involved, and which change the composition of microbial community, which make microbial communities undergo succession. The other aspect of this time 1 is not equal to time 2 is, for example, you go to Haridwar and you sample the wastewater treatment plant, okay, and you are like, already Haridwar wastewater treatment plant has this much problem, has this much emerging contaminants, and has this much of DO, this much of BOD. So you get the information for the water from this Haridwar wastewater treatment plant. Now you go again next day, and not only that, not only the basic information, you also do microbial community as an analysis. So you know species 1, species 2, species 3, so on and so forth, right. Now if you go next day again to Haridwar wastewater treatment plant and collect another sample, then it is very likely that the microbial community will not resemble this, So this is a very important point, that the time of the sampling makes a big difference in the microbial community I am hoping to record. So this is a major hurdle for ecological studies, and one simple way out of this is to do replicates, or to sample everything at the same time, and to have identical conditions, which is very challenging when we are trying to do field related work. The next hurdle for ecological study is 99% of bacteria are non-cultivable. So basically the kind of bacteria that you can culture in the lab, like tuberculosis, and you can culture so many other Pseudomonas aeruginosa, Staphylococcus aureus, all these can be cultured, so we know a lot about them thankfully, but 99% of bacteria are unculturable. We have absolutely no idea who they are, what they are. And if nothing else, this I hope definitely serves the purpose of humbling us into realizing that very little is known. So now let us take a recap and find out why 99% of microbes are uncultivable. Obviously the simple reason is because the environmental conditions are very different here. So basically this is the environmental conditions in the lab are different. For example, let us say there is a scientist, a very renowned German scientist, who goes to hot water springs and lakes and vents, thermal vents, to collect samples so that he can find out the bacteria. Now let us say he gives me his hot water sample in which there are archaea and there are extremophiles growing, and I take them to my lab and I try to grow them on the broth. Now here's the thing, my broth is at 25 degrees Celsius. The bacteria, they were living at 98, 85, definitely above 70 degrees Celsius. How will they survive in the plate? That is not possible. So the environmental conditions, when they change, they can, because the environmental conditions in petri dish are very different from environmental condition where these microbes actually grows. That's why 99% are uncultured, cultivable. The other reason could be that they like to exist in microbial communities. This is another reason, because microbes, they prefer to exist as communities. They do not like to grow individually in the lab, and only few of them can. That's only few of them are culturable. And then there are many other reasons that we have not completely understood yet. So because we can not compare two time intervals of ecological samples, so we need to do replicates, and we need to rule out all other factors. We need to collect lot of samples, generate lot of data. And because 99% of bacteria are uncultivable, we need to find culture independent sequencing techniques. And obviously metagenomics shines as the most popular culture independent sequencing technique. Now let's look at the cost of sequencing. In 1990, sequencing was very expensive, and following Moore's law, and even better, the cost has reduced a lot, and it's even less now. Alrighty, now as the price is reducing, as the need is being more and more appreciated, obviously the trend for number of publications in leading databases is like this. So in 2000-2005, there were very little papers on soil metagenomics. In 2006-2009, we had considerable number. In 2010-2015, we just shot up. So there's a lot of scientific interest, scientific community's interest. Now let's look at metagenomics again briefly as a recap. So what do you do in metagenomics? You take your sample. This is your, let's say, marine microbial community, so it grows in ocean, and they have some sequence of their genetic material. So first step is you extract DNA. So this is step 1, total DNA extraction. And after you have done DNA extraction, you might go for shotgun studies for all environmental genes, and after doing shotgun, you go for the second step, which is DNA sequencing. So in this case, you do the DNA sequencing. When you get the data, you identify the common genes within a community. For example, I noticed that I put very high pH in some place, and still there are microbes growing, but I put high pH so that they don't grow, but they are growing, and I want to find out what makes them grow at such high pH. So what I do is I can sequence the DNA, find out for the similarities. What are the similarities between all microbes across the globe that grew at high pH? Once I get the similarity, I can be more sure that already this might be something new, some new microbe, and I need to pay attention. And after you have got the sequences, what you do is you identify common genes within a community. So once you get common genes, it makes analysis very easy, and then you identify genome contents favored by current environmental conditions. This is a very important sentence. Next is protein annotation. Okay, now you have sequences, you are pretty sure where they are coming from, you know how to deal with it. Next step is very difficult, it's called protein annotation. Again, you use metagenomic studies as a tool to answer broader ecological. So you use whatever information you have, whatever tools you have in your hand to answer broader questions on ecology, and here it says evolutionary, but just environmental microbiology. Alrighty. So let's look at metagenomics, what metagenomics look like. Metagenomics we can do for functions, so basically kind of functional metagenomics, or we can do microbial composition, which is basically telling me who is present. And function is what is happening. So if you are doing microbial composition assay, which are the most typical assays nowadays, you want to find out biodiversity. So you want to look at alpha diversity, you want to look at beta diversity, potentially even gamma diversity. These are not gamma protein bacteria, alpha, beta, delta, epsilon. These are very simple, kappa and all that, but this is very simple. So what you can do is you can look at biodiversity. How are the sequences in this sample different from the other sample? So for example, samples from Mumbai landfill different from Delhi landfill. This analysis can be done here. Here, when we are doing better biodiversity, we can also do in sample diversity. Are microbes in Mumbai landfill more diverse than the ones in Delhi landfill, or is it the other way around? So all this will come under biodiversity. We can also look for specific bacterial groups. Already I am doing cellulose degradation. Tell me what kind of Clostridia are present. I am looking at only Clostridia and I can look for them. Next, we can find a relation of microbes and environment. So whenever I take sample from an environment, the microbes that are thriving in that environment are the ones that are very well suited for that environment. So what I can do is, because I know the environment, because I picked the sample from there, I know its temperature, I know its humidity, I know its conditions, I can pair up, okay, in this kind of condition these microbes grow well. This information is very, very helpful for commercial purposes and also for research. Next, what I can do is, using my microbial composition, I can look at reconstruction of putative metabolic networks. Now, if I am looking at function, with function I can understand the metabolic networks very much similar like this. I can look at specific functions. For example, if I am doing cellulose degradation, I might look at the functions that talk about degrading starches, talk about degrading cellulose, and I can also target specific genes. Already, this is perhaps going to be the second last slide here. The question is, already we need to do metagenomics, we need to do sequencing, because these are the conditions that we face as environmental engineers. We cannot culture 99% of bacteria, so there is no point trying to culture anything. So let us do sequencing. Now when we do sequencing, we need internet. Why do we need internet? Well, because more often than not our own PCs are not sufficient, they are not built for and they are not sufficient to do bioinformatic analyses. So we need to have our institute computer that you can use, or in case if you have a good computer at home, you can definitely use that. And once you have used that computer, you upload the data to NCBI, NCBI verifies it, might send it to GenBank, and then you get published and stuff. Why is this important? Look here, the omics project like metagenomics, like proteomics, metabolomics, all these omic projects, and we have talked about omics before, it means all. These omics projects and the information associated with them involve a huge amount of data that is stored on computers all over the world. So if we are connected to internet here and we are doing metagenomic analyses, our data will be shared by other computers. So it will not be very taxing on our own personal computer. It is impossible to maintain up-to-date copies of all relevant databases within the lab. Access to the data is via the internet. It is not possible that we have up-to-date copies of everything, every database present in the world within the lab. So people should be able to access it online. For example, recently my colleague from Virginia Tech informed me that they need access to the Linux computer that I was using when I was doing my PhD. Why? Because, well, we sit at home and we do the work. So for that reason, we do need to connect to internet. And then this is the last slide. Let's say you are here connected to internet. This is your database storage. And then what happens? You send a request. For example, let's say you have a file. In one of the lectures we will be doing this, and you send, please align this file with your database, tell me what it matches best with. And what this will do is, it will go for gene matcher, it will go for alignment, for BLAST, for MEME, for MAS, for pattern finder, and after it has done all that analysis, it will get back to you with information. Alrighty, the last slide for today. The current bioinformatics market, this is in USA by the way, is worth 300 million per year. Prediction is it will rise up to 2 billion per year in 5-6 years. There are at least 50 bioinformatics companies at time of making this presentation in USA. So there is a lot of scope even in India. We have talked about it, scope in public health, scope in general health, in well-being. Also lot of people who are not able to exercise, but they need to know how their body is changing with it, and then they generate a lot of data. Then they need to make sense out of this data, and to make sense out of the data that they are generating using wearable sensors because of their health issues. That data analysis also is done by bioinformatics. Alrighty students, this is all for today. In next class, we will go more in depth about bioinformatics, trying to understand what tools are available to us easily and how we can use them best. Thank you.